Greetings and welcome to Nursing 103. This is week 1. We'll be covering sections of chapters 40, 41, and 42, microbiology, medical asepsis, and infection control, cornerstones of basic nursing care. You will note that this is a rather large reading assignment. This is the largest reading assignment of the term. We have 2 normal class weeks to cover this, although you can get it done obviously much sooner than that. The infection control, medical asepsis, and microbiology homework is on my IoT, and it's under the online exam. It is not only okay to use your book, it is really required that you use your book to answer these questions. If you have any questions regarding that, you can email me directly or post a question in the comments section under the video. Okay, well, let's get to it. So what is an infection? Right? An organism invades and multiplies in body tissues and can cause injury to the host. We get an infectious disease if we have a chain of infection that continues from one host to the next. We need to break this chain. We are seeing currently this is an issue with social distancing and self-isolation, trying to break the chain of the COVID-19 pandemic. So for normal course, you have an incubation period. Okay? Pathogen enters the body, and the first symptoms of illness appear, right? That time between when it comes in and the first symptoms, that's our incubation period. Prodromal stage, initial symptoms might be a sniffle, might be a sneeze, to the more severe symptoms. The full stage, right? Acute and specific to the type of infection. So consider if I had a respiratory infection, okay, let's just say influenza A. I would have symptoms, body aches, fever, mucus production, cough, but I would not have a rash because that would not be specific to the type of infection, if this makes sense to you. And finally, the convalescent stage. Yay! The acute symptoms of the infection are over. The immune system has done its job. The medications have done their job, and the person begins to recover. But what can develop an infection? Okay. What do we need? Okay. So a portal of entry. These could be through mucous membranes, through breaks in the skin. Right? The number of microorganisms. Was it one virus that got in, or 1,000? Was it one bacteria that was easily overwhelmed by the body's immune system, or was it 1,000 bacteria that got a foothold and started an infection? The virulence, how likely it is to cause disease, and then the host's resistance. What condition is the host in? Is their immune system in perfect shape? Is their immune system compromised? Are there breaks in the nonspecific defenses, like intact skin? Let's look at some basic terms here. So, microorganism. It's an individual living animal or plant, single-celled. Right? Happily, most do not cause disease under normal conditions. It's when the conditions get abnormal, or you have higher numbers of microorganisms, that they can begin to cause disease. Right? Endemic. So endemic to the body or in populations. An example here, we always hear about staph infections, but we have staph present on our skin. Most of the time does not cause an issue. With the break in the skin, though, with the right set of conditions, then you can get a staph infection. Okay, staph would be endemic on the skin. In a population, you may have a disease or disease process that is common in a population. Say, used to be, polio was endemic in certain parts of the U.S. Not anymore, thank you, Jonas Salk. But it was there; it was present all the time. Right? And a pathogen, pathogenic, harmful or disease-producing. So a pathogenic microorganism is one that can produce disease or cause harm. Note I've got it in red here. A nurse is responsible for taking steps to prevent disease transmission, okay, to prevent the spread of infection. That is a number one job of nursing. We are here to protect our patients. And I'm going to give you one big guess as to what the first step in breaking the chain of infection might be. We'll come to that in a minute. Endogenous microorganisms. Microorganisms present within the person's body that cause infection. Okay, so within. Right? So Staph aureus. So on the skin, right? Staph aureus, Staph epidermidis, the not the methicillin-resistant Staph aureus. It may be present, but that is definitely a pathogenic organism. Exogenous microorganisms come from outside. Salmonella. Sorry, voice is getting a little dry here. Hold on. So Salmonella, Clostridium tetani, Aspergillus. These are all from outside the body getting in, and they can then cause some harm to the host. All right, let's pause for a couple of minutes here. You can go ahead and hit pause, take a break, take a drink, stand up, turn around, and then start up again. We'll come on to characteristics of microorganisms. So metabolism, the ability to utilize nutrients. Okay, so just like the organism they are trying to inhabit, us, microorganisms need food. They utilize food. We are their food. They're able to reproduce, band together, form little colonies. They get larger. Okay. They become irritable, so they respond to their environment. So if there is a heat source, they may move away or they may move towards it, depending on the microorganism. But they respond to the environment. They can move from place to place. And they have some form of protection. Spores, like fungi, mutations to adapt to threats in the environment, and part of this, the development of drug-resistant strains, what we call the superbugs, if you will, the microorganisms that are now resistant to all of our standard treatments, or nearly all of our standard treatments, and we are running out of options to treat these superbugs. Obligate aerobes, okay, aerobes and anaerobes. Got those in red there. Obligate aerobes need oxygen. Right? Just like you or I, we need oxygen to burn glucose, to create energy, to go on with growth and development. Obligate anaerobes, no oxygen. They will die in the presence of oxygen. Nutrients. Got to have building blocks to build things, right? So just as we require the building blocks for proteins, which are then the building blocks of all of our cells, the microorganisms require building blocks to build themselves. Parasites, well, those feed directly on us or from us. And then saprophytes live off the dead parts of dead plants and animals. So if you, it's not a microorganism, but if you think of maggot therapy for wounds, where sterile maggots are placed in the wound and they eat the dead tissue, they are then saprophytes. Okay, here we go. You will hear this phrase throughout the program, at least in all of my classes, and you'll hear it over and over again, so become familiar with it now. Life, as we know it, generally exists in a fairly narrow range of temperature and pH. That's life as we know it. There are some outliers in the depths of the ocean, but a fairly narrow range of temperature and pH. Now, temp, of course, lovely for pathogenic microorganisms. Normal body temperature is where they thrive. Okay, well, that's unfortunate for us because normal body temperature is where we thrive as well. pH, microorganisms survive in an environment, so our pH is neither too acidic nor too alkaline. We as an organism run slightly alkalotic, right? The pH scale goes from 0 to 14. 0 is completely acidic, 14 completely basic. We run in these, and 7 is neutral. We run at the 7.35 to 7.45 range. Just slightly alkaline. Some microorganisms need light for growth. Others need darkness. Ultraviolet rays, UV light. The majority of the really nasty UV light is filtered out by our ozone layer. But the super short wavelengths of ultraviolet light disrupts DNA and causes the microorganisms to break down. Also the same thing that causes skin cancer in humans. That overexposure to the UV. Moisture. Well, what is it? 2 weeks without food. 2 minutes without air. You need, and 2 days without water. I think that's it. Have to look that one up again. That's been a while. So, without water, it's a major component of all of our, major component of all of our tissues, all of our body fluids. Water is necessary for life as we know it, and so microorganisms still have to have some moisture available. Now, we're going to kind of fluff through this a little bit because we won't be growing the cultures ourselves, but as an understanding. Okay, when we take a specimen to send to the lab, down to the micro lab, right, they grow them in cultures, very controlled conditions, right? In a tube, a petri dish, in the culture medium, it's sterile. We'll start with that because you're trying to grow specifically what it is that you sampled, not whatever happened to be growing in the lab at that time, if that makes sense to you. But we have different types. We have liquid and solid. Anyway, either one that you use, still we're inoculating that, or the lab is inoculating that with whatever we sampled to see what grows. So that we can identify what the microorganism is that is causing the illness. And then we can also figure out what we can use to combat it. Here's the culture and sensitivity. So the culture, which we've just talked about, grows the microorganism. And then the sensitivities, dots of different types of medication, types of antibacterials, are placed on the medium, and let's see which ones, because around those dots the microbial colony dies off, right? And that determines which one we can use to treat the infection. And it also sees, okay, are we being effective with this therapy, or has the microorganism begun to develop a resistance? Very important in monitoring is monitoring for new types of antibiotic resistance. So, the culture and sensitivity report. What are we looking for? What did we find? And what is going to kill it? Right? Preliminary or final, because these reports take some time. There's initial report or initial results that come back, and then the final determination after 24 or 48 hours, whatever the test might be. It tells you what's growing and what it's susceptible to, right? So culture and sensitivity, culture and susceptibility, those are the same CNS. C and S, not CNS. C and S. And what will be effective against it and what will not be effective against it. This also helps us monitor for the development of drug-resistant microorganisms. Viruses. Here we go. In light of our current situation, the COVID-19 pandemic, and the reason for this online presentation. So viruses are protein-covered sacs that house all their genetic material, DNA or RNA, depending on the type of virus. But it requires the mechanisms of another cell to reproduce. So when the virus enters the body and it enters the cell, where the microbiologist created the culture medium to grow the bacteria, well, the cell becomes the culture medium for the virus. I have things like pox virus, smallpox, adenoviruses, picornaviruses or coronaviruses. We hear coronavirus and we get all choked up, but there are a number of human coronaviruses that we are in contact with that don't cause much more than a runny nose. The COVID-19 or SARS-CoV-2, I believe is the other name for it, is just the newest and scariest. So, virus requires a cell to reproduce in. Fungi. Great on pizzas. If you like beer, you love fungi. Mycosis is an infection caused by a fungus, right? Yeasts. Candida albicans. Not a fun one. This is not your beer yeast or creating bread. This is the one that causes thrush. Oral, vaginal thrush. This is your yeast infection. Okay? And then molds. We have a number, from the lovely green mold that grows on the toaster, that science experiment that used to be leftovers in your refrigerator. Some can be beneficial, as we find that some molds inhibit the growth of bacteria, hence penicillin. However, they can also be quite pathogenic in humans, like histoplasmosis, causing histoplasmosis, I should say. Pardon me. So, bacteria. When we think about the world around us, what we don't see, living on every surface, floating in the air, just waiting, millions and billions and trillions of bacteria. Now, illness-causing, like prokaryotes. Right? They're very small. They have a cell envelope, right, or the skin, if you will. Appendages, their arms and legs, or cilia, flagellae, those little appendages that allow them to move. And then the cytoplasmic region inside, where all of the life processes are going on inside that bacteria. much like a human, only on a single cell. So, reproduction and survival. Through conjugation, cell-to-cell contact, genes transferred by a virus, as in transduction. Transformation, a bacterium will pick up DNA from the environment that has been released by another bacteria. This is how resistances to antibiotics are often transmitted, as they share DNA. Certain bacteria, they can spore up, like fungi, form a hard spore that is very difficult to break through. An example of this would be Mycobacterium tuberculosis. It's a bacterium, but it also has spore-forming capability. This is the causative agent for tuberculosis, pulmonary tuberculosis, that we see in human populations. And these can survive some pretty extreme conditions, which is what makes it so hard to treat tuberculosis. Temperature, water's boiling point. Recall we said life exists in a fairly narrow range of temperature and pH. Everything except the spore formers will die at boiling point, 212 degrees Fahrenheit, at water's boiling point. Which is a plus for us. When you see in the news, there's a boil water order. This is being done. There's a bacterial bloom or an algae bloom. Well, an algae bloom may not be something you want to boil water for, and we'll cover that here in just a second. But to boil the water to make it safe to drink by killing off the pathogenic bacteria. Here is where the boil water order may not do you any good. Toxins. So toxins, toxic, poison, right? Endotoxins, they're part of the cell walls, right? So they're within, right? So when the cell dies, it's held this toxin inside. The cell dies, breaks down, it releases that toxin into the bloodstream. So here, well, it's 2 years ago now, we had the algae bloom up in Detroit, and we had the great water shortage because of the endotoxin that was being released, cyanotoxins that were being released from the algae bloom, right, which could cause some pretty nasty effects in humans and animals. Okay? These are also the cause, endotoxins are also the cause of some potential complications of antibiotic therapy. So we have bacteria A, and we treat it with an antibiotic, and we give the first dose of the antibiotic, and it begins to break down and kill off bacteria A. Okay. With the dying of that bacteria, as it breaks down, it releases these endotoxins, and it's very common in some of these infections to see that after the first dose of antibiotics, the patient's blood pressure will drop, and it's as an effect of these toxins. So it's something that you want to be monitoring when you start your patient on antibiotic therapy, is you're looking for changes in vital signs, blood pressure, not just allergic reactions. Right? Exotoxins Um, you could think of this almost like a spider in venom. The bacteria manufactures it and then excretes it out in the surrounding tissue to break it down so that it can eat. Now here we go. To be pathogenic, to be disease causing. Okay, that's pathogenic, disease causing. Have to be able to colonize and invade and then colonize the host, right? They have to be able to resist the host's defenses and be toxic to the host. That's to cause disease. Otherwise, they're just little critters living on or inside us. So consider Bacillus anthracis, anthrax, Klebsiellas, gonorrhea. Right? These all colonize. They increase in numbers, resist the defenses, and become toxic to the host. And here, if we were face to face, would be where I get out my soapbox and stand up on it. Drug-resistant microorganisms. We are all very focused on a particular virus at this moment, which is no doubt a threat, but a longer-term, greater threat to our survival as a species are drug-resistant microorganisms. Are antimicrobial agents that we use, antibiotics, if you will. We are rapidly developing microorganisms that are resistant to the tools we have to fight them. Well, okay. So what's the big deal? We need to just make a new antibiotic, right? New antibiotic development is very slow. Thus far, not any really good candidates. So we need to really work on utilizing our infection control procedures to stop the spread of these infections. And part of this, as we're going to go through in just half a second, has to do with patient education. Okay. So, what do we teach our patients to prevent the development of new drug-resistant microorganisms? First and foremost, unless there is an allergic reaction, take their antimicrobials, their antibiotics, only as prescribed, meaning for the entire course of the antibiotic. If it is 3 pills a day for 10 days, they take 3 pills a day for 10 days, and that they do not stop taking it just because, well, their infection, they feel better, so they don't need the antibiotics anymore. Teach them to take them as prescribed and the necessity for doing it as prescribed. Not to share their drugs with others because they're prescribed the course of antibiotics for themselves. Now, you may see this sharing of the antimicrobials in cases socioeconomic challenged patients, low income, no insurance, undocumented, anything that impairs their ability to seek medical care. Right? One may get it and then share their antibiotics with the rest. Well, what we're doing here is we are creating a drug-resistant microorganism, because the old saying, what doesn't kill you makes you stronger, is really applicable in this case. What the short course of antibiotics does is it knocks off the weakest of the bacteria, allowing the stronger to grow and take over. Right? So don't share them with others. The last piece in this, do not use antibiotics for viral infections, because antibiotics do not work on viral infections. Those would be antivirals. Those are a much rougher class of drugs, and they are not indicated for everyone. In some cases, the effects of the antiviral would be worse than the disease that they would be taking it for, like the common cold. Right? Antibiotics do not work for viral infections, and we have all known that that person who has taken their child to the emergency department and screamed and gnashed their teeth and wailed and threatened to sue because the ER doctor would not prescribe them or their child antibiotics for their cold or for their flu. And the reason that they would not be prescribed those antibiotics for the cold or the flu is because antibiotics don't work for viral infections. Okay, now that we've talked about infections and microbes and all of the ways that things can spread, now we're into the heart of the nursing matter, asepsis. Now we'll find there's a difference between medical asepsis and surgical asepsis. First off, terms here. Asepsis, minimize or eliminate pathogenic organisms. That's simply the term. Medical asepsis, reduce the number of microorganisms to prevent or reduce transmission of microorganisms from one person to another. This is what we're using in general bedside care, general nursing care. Right? This is, we asked a question earlier, hand washing. Hand washing, by the way, is the number one way to limit the spread of microorganisms from one person to another. Wash your hands. That's all over the news right now. Wash your hands, use hand sanitizer. So, medical asepsis starts with washing the hands. Surgical asepsis. Now, here we go from reducing to destroying all organisms, microorganisms, but this is used only in certain situations. Again, this starts with washing your hands. Just the hand wash is even more in depth. Now, due to the time involved, we cannot use surgical asepsis with every patient contact. would be wonderful if we could, but we would get about 4 patient contacts in a shift if we were to do this. Nosocomial infections. Where is the best place to get an infection? I want you to think about that for a second. The best place to get an infection. And at the end of the slide, we'll talk about that. So, nosocomial infections can lengthen a person's stay in the healthcare facility. Okay, nosocomial, healthcare acquired, right? You got it at your clinic, your inpatient treatment, long-term care, hospital, right? Lengthens a person's stay, increases the cost of treatment because now you're treating the patient for something that they did not have when they came in, and can kill the patient. We, as healthcare personnel, are major vectors here. Okay? We can cause the transmission of a nosocomial infection by not using appropriate prevention techniques. We increase the patient's possibility of exposure to pathogens. Cross-contamination, because we don't just have one patient, we have multiple patients. And not following standard precautions. We'll come to standard precautions later, but very basically, standard precautions, it's in the name. These are standard. These are what we take with all patients, whether they are here to drop off paperwork or here to have surgery. Everything based on standard precautions. Okay. So, what's the best place to get an infection? In a healthcare facility. Why? Because that's where all the sick people go. Think about that for a second. You are training to be ground zero, where all the sick people are going to go when there's an outbreak, as what's happening right now. Okay, so most common nosocomial infections: genitourinary, UTIs, about 40%. That's a lot. Bacteremias, 14. Surgical site infections, respiratory infections, and GI infections. Surgical site infections, now GU infections, these are all higher. What am I kidding? These are all higher than they really should be. Okay? And these have also led to changes in how facilities operate, how Medicare and insurance pays. There are conditions that if your patient comes in with this condition, or if this patient develops this condition while they're in your care, that Medicare won't pay for, Medicaid won't pay for, and the facility eats the cost, like catheter-associated urinary tract infections, like surgical site infections, respiratory infections if they are associated with the use of a ventilator. There are things insurance will not pay for, and your facility eats the cost. And this is unfortunately the way now that insurances are getting the attention of facility administrators saying, hey, this has to change. And this really does need to change. Because with all of these, nurses are involved with all of these situations, and we need to be stepping up our game to decrease these numbers. Okay, so more common nosocomial infections. Everyone's friend, MRSA, MRSA, Staph aureus. So methicillin resistant to a lot of the cillins. Okay? VRE, vancomycin resistant enterococci. All right. So when you got MR, methicillin resistant, and VR, the scariest of the two would be the VR. Vancomycin is one of the big guns that takes everything out, and when it's resistant to vancomycin, we begin to have troubles treating the disease. Right? ESBLs, right? Penicillin-resistant strep pneumonia, right? Now, this, though more common and older, can happen in younger patients as well, but penicillin-resistant, so we have to treat with a different class of antibiotics. And then epidemic MRSA, when you have a big outbreak of MRSA causing disease. Now, does this mean that we do not have, if we have MRSA in the population, is it epidemic? No. In healthcare, do we likely come in contact with MRSA on a regular basis and not know it? Yes. It's when it gets a foothold and begins to cause disease and dysfunction. And in epidemic MRSA, as we're seeing with our pandemic virus situation, epidemic MRSA, when it's affecting a large number of people in an area, then it becomes a greater problem for treatment. The virus causing SARS, the coronavirus. Not to be completely confused with our current novel coronavirus, or SARS-CoV-2, or the causative agent for COVID-19, severe acute respiratory syndrome, NDM, CRKP, NDM, CRKP not seen quite so frequently. Of course, and on this, a lot of focus is being given to the new coronavirus. And you'll note something that's not showing on here, our influenza, which can run rampant through facilities as well. HA-MRSA, hospital-acquired methicillin-resistant Staph aureus, or healthcare-acquired. So this is the patient that came in without MRSA and contracted MRSA in the facility. And now it is healthcare-acquired MRSA. And how did they get it? Usually through the contaminated hands of healthcare workers. That is us, folks. That is us. How could this have been blocked? By breaking the chain of infection, by washing the hands and using standard precautions. Okay, so what things can increase the risk for developing a nosocomial infection? Well, number one, a compromised immune system. Disease conditions. AIDS, acquired immune deficiency syndrome. It's kind of in the name. Cancer. Cancer affecting the blood or the bone marrow, which directly impacts the immune system, as in the bone marrow is where the white blood cells are produced that are key components of our immune systems. Aplastic anemia. Now, normally when we think of anemia, we think of a deficiency in red blood cells. However, aplastic, the blood cells are not being made. This may also fall over into the white blood cells are not being created. Chemotherapy and radiation, treatments for cancer and other conditions can cause a heavy impact on the bone marrow, again decreasing the production of white blood cells and increasing the risk for developing an infection. And then last, corticosteroids. Now, we say overproduction of corticosteroids. This could fall under two situations. One, the patient has a condition that causes the adrenal cortex to overproduce these compounds, okay, these hormones. But there's another piece of this. So if my patient is receiving corticosteroids for treatment of another condition, this is a de facto overproduction of corticosteroids in the body. This can weaken the immune system, again leading to an increased risk of developing an infection. And when you're in a healthcare environment, a facility where, as mentioned before, all the sick people go, this just increases the risk for developing an infection. Breaks in the skin. Say, you have surgery, burn injuries, more than just a superficial, more than just a first-degree burn, but a deeper burn, where the surface of the skin is no longer intact. That opens up a portal for infection. The presence of a PICC line or an IV. So these are catheters going into the body, into veins. In the case of the PICC line, it's going into a central vessel, which is a great way to deposit bacteria directly into the bloodstream. Another risk factor for clients in healthcare facilities, the presence of urinary catheters. Your Foley catheter. These are associated with high numbers of urinary tract infections, and it is also one area that is really actually quite easy for us to decrease the risk for developing infections off of these just by proper catheter care and not using Foley catheters unless absolutely necessary. So when we have a lowered resistance, right, trauma, so physical trauma, emotional trauma, severe stress, fatigue, these are all things that can overtax the immune system. Now, for stress, just last slide, we were talking about the overproduction of corticosteroids. Corticosteroids are released when the body is under continued stress. So, lowering resistance. Fatigue. Pre-existing disease. So if you're already sick and you enter an environment with a high concentration of pathogenic bacteria, your resistance is lowered because your body is already tied up fighting off the disease you've already got. If that makes sense. Generally poor health, or if you're frequently ill. Now, frequent illness can point to immune issues beyond what the scope of what we're talking here. Age, very old or very young, are more susceptible to illness. The very young systems aren't as well developed, and the very old systems are getting worn out. Inactivity, poor nutrition, because you need energy to fight disease and heal. If your nutrition is not up to par, not up to your body's requirements, that can impact your ability to fight off infections. Inadequate hydration. Water is life. Invasive therapies like IVs, like surgeries, Foley catheters, urinary catheters, or immunosuppressive situations like we talked about before with medications to suppress the immune system, chemotherapy, radiation. In some cases, we would be treating a condition by suppressing the immune system. For example, a transplant patient, someone who's had an organ transplant, will be on anti-rejection drugs. And they will be on anti-rejection drugs for life. And what the anti-rejection drugs are doing is tamping down the immune system to keep your body from kicking out the brand new organ that has just been implanted. And frequent or inappropriate use of broad-spectrum antibiotics. There's a good one. So frequent or inappropriate use, broad spectrum, because we're developing resistance, okay, in the bacteria. And then inadequate primary and secondary defenses in the body. So an impaired immune system, breaks in the primary, so skin and mucous membranes. If skin and mucous membranes are not intact, you have portals for infection. So this one will take a little bit of explaining here. So previous exposure to antimicrobial drugs, reducing the normal flora. A good example of this would be Clostridium difficile, C. diff. This is present in the GI tract, as are a number of other bacteria, microorganisms that live in our GI tracts. When we take antimicrobials, as we go back here a little bit earlier, talked about the culture and sensitivity. Different bacteria sensitive to different drugs. If we are taking antimicrobials, realize that they are not just taking out the bacteria that are causing the pneumonia. They're affecting all the bacteria that they will affect all the way throughout the body. Okay. Now, in the gut, you have your normal flora that is usually kept in balance. Consider these are different colonies of bacteria that are competing for the same supplies, the same nutrients. So we have bacteria A, bacteria B, and bacteria C, diff, and they are all competing for the same stuff. As long as they are all competing, none of them overgrows. Then we take an antibiotic that knocks down bacteria A or bacteria B or bacteria A and B, leaving bacteria C all this extra room now, and all of these resources that it could possibly exploit, and it overgrows, gets out of balance, and creates a super infection. So in this case, the previous exposure to antimicrobials takes things out of balance and allows one microorganism to take over, causing an infection. Repeated hospitalizations. Again, best place to get an infection is the hospital, because that's where all the sick people go, as they should. Old or young, and then previous infections by multi-drug resistant organisms. Because we know that bacteria have a tendency to share their DNA. And with multi-drug resistant organisms, this gets rather scary for healthcare professionals, because we see more and more microorganisms becoming multi-drug resistant. And we lose bit by bit pieces from our toolkit that allow us to treat and care for these patients. Now here's the biggie. Nursing staff. Okay, consider if my patient gets pneumonia. Okay, my patient is affected. Now, the nurse gets pneumonia. How many people are affected? It would be every patient that nurse is going to treat during that time. Do we run a higher risk than the general population for being exposed to diseases? Well, yes. Tuberculosis, hepatitis, COVID-19, or SARS-CoV-2. We run a higher risk for exposure because we are the ones caring for these patients. Hepatitis, A, B, C. Generally don't see A as much in healthcare, but we're vaccinated for hepatitis B. We're at a constant risk of exposure with hepatitis C because many patients have it and do not know. HIV. So we need to get the immunizations, carefully wash our hands, use standard precautions, and above all else, use your brain. If you have sharps out, you handle all of those sharps as if they are already infected with something nasty. Right? Strict sharp safety, standard precaution, hand washing, hand sanitization, and staying on top of your coworkers as well. Because we are in the front line treating patients who are sick with infectious diseases, and we do not need to bring those home to our families. and also impact all of the patients that we might have treated, but now we can't. Coming down to the home stretch, the infection control plan. Because we have infections, we need to control them, therefore we need a plan. Great place to start is with an infection control committee. A surveillance and reporting system for nosocomial infections, an employee health program. Right? These are all parts of a good infection control plan. Isolation policies, infection control education for employees, and not just done at the time of hiring, but frequent, at least yearly. Environmental sanitation controls, environmental services. Oh, wait, environmental services, aren't those the housekeepers? Yes. Housekeeping or environmental services are a really big part of infection control in facilities. Got to have an available micro lab. Why would I need a microbiology lab? Well, if I'm trying to track and control infections, I need a way to identify those infections. And then infection control procedures for client care. All great parts of an infection control plan. Let's look first at the Infection Control Committee. It's a central reporting place, reporting infections. Because in a facility, you may have, or in a facility or in a healthcare system, you may have multiple clinics, multiple buildings, houses, towers, depending on where you're at, what type of facility is. And in those, you might have different wings and different floors. And they all begin to develop their own little specialty. And information tends to silo in those areas, right? Because they're all busy taking care of their patients. And they're all busy taking care of their patients. So at the Infection Control Committee, there's a central point to report infections that develop. Okay? Also a place that the investigation of these cases can begin and operate from. A central point, not associated with any one building, wing, floor, division, whatever. Central point for the infection committee, infection control committee, to start investigating. This helps us to determine the cause of the infection. Because now that we found it, and we know how this infection came about, how it was spread, then we can design policies for infection control. We can better train our staff on infection control. And with that, we can break that chain of infection, hopefully, and prevent it from spreading into other parts of the healthcare system. And the last bit is work to prevent further recurrences. So we found that one area was getting a lot of urinary tract infections, and we discovered that it was Nurse Jones that wasn't using proper technique inserting Foley catheters. We retrained Nurse Jones. Nurse Jones now has no more cases of urinary tract infections in the patients that Foley catheters were placed. We can now use that training that we used for Nurse Jones to train the other staff to prevent further recurrences. Watchfulness for HAIs. So we have that. It's a central reporting point. Oh, we got a healthcare acquired infection. Okay, where is this? It's just one. Great. Oh, nope, there's another one. So looking out for, all right, where are they at? Looking for the infections, investigating the cause, compiling the statistics, because unfortunately, statistics run a lot of treatment funding decisions. We do the staff teaching, serve as a liaison between the facility and the community. So you'll note in the current situation, hospitals, their infection control officers working with the community to relay information about procedures to get into the facilities, right? Limiting the decision had come down to limit visitors, right? These are to slow the spread of the infection or to hopefully stop the spread of the infection. Right? And a good infection control committee should also have an eye out on the surrounding community and the world itself. And there we have the end of week 1. We will be experimenting with different formats as we continue to navigate this. We'll see what week 2 brings. Any questions? Again, email me or drop them in the comments below, and I will answer your questions as I am able. Should be by Wednesday.